Hi, welcome back to Mitosis. All right, we just finished talking about prophase and prometaphase. So by this point, the cell has just exited G2 in interphase. Remember, interphase is G1, S, and G2. And it's copied its centrosomes, which are the microtubule building centers in your cells, if you will. And now the DNA, as it enters prophase, is being told, okay, we're shutting things down, everyone condense, get nice and tight. So the chromatin is going from this, 6-foot thing going into this tiny little package that we know as our chromosomes. So the chromosomes are getting nice and condensed. As they enter prometaphase, the nuclear envelope is breaking down. The microtubules actually now have access to their kinetochore region, and this is where we left off. So now, after prometaphase, enters metaphase. So we have prophase, prometaphase, which is your in-between stage, and now metaphase. So big letter C is metaphase. So during metaphase, this is an excellent stage of mitosis. This is when I want you to imagine we're only working with 2 chromosomes in our pictures here, but there's really 46, right? So during metaphase, here's what's happening. Your cell, the nuclear envelope is gone by this point. You have your centrosomes, and we'll draw the microtubule in a second. But during metaphase, all of the chromosomes are lining up along what's called the metaphase plate. It's this invisible line that goes right smack dab in the middle of the cell. And along this invisible line called the metaphase plate, the chromosomes line up along their centromere. Oh, yuck, I'm gonna redraw that chromosome. Sorry, I like pretty chromosomes. And gotta stay somewhat consistent on how this looks. All right, that's doable. All right, so we got our chromosomes there, and they're lining up along the centromere. And this is wonderful how this happens. If you want to split something evenly, it's kind of nice to be able to line them all up along their center line, and then you can tear them apart. That's exactly what's going to be happening here with our chromosomes. Excuse me. So the centrosomes that have now created these microtubules that are latching on to the kinetochores, and you have your non-kinetochore microtubules. I'm keeping the drawing very simple so you can see things. But what you need to remember is that if I'm a chromosome, on one side, one of my kinetochore microtubules belongs to the centrosomes on this side. One of the other kinetochore microtubules belongs to the centrosomes on the other side. How they do this, we still don't understand. I find it fascinating that this set of centrosomes knows to latch onto this side of the kinetochores. This set of centrosomes know to latch onto that side of kinetochores so they can pull them apart equally. You're kind of visualizing what's going to be happening with metaphase now. This is actually the longest phase of mitosis. It takes a long time for them to get organized, and it's about 20 minutes long, which in the cellular world, with the way things work pretty fast inside cells, that's actually a decent amount of time for mitosis, for this phase in mitosis. So during metaphase, here are the big takeaways about metaphase. During metaphase, it's the longest phase, which is pretty cool to know. But the centrosomes are obviously migrated to opposite poles by this point. They're definitely there. And the chromosomes line up along this invisible line called the metaphase plate. And they line up along their centromeres, the inside part where the 2 sister chromatids meet. So the chromosomes, which are made of 2 sister chromatids, right? The chromosomes line up along the metaphase plate. It's a completely invisible line. By their centromeres. Imagine 46 of these just lined up perfectly, and everybody's attached at this point. So by this point, every chromosome has a kinetochore microtubule from either side of, from both sides of the cell. Has a kinetochore. So one from one side, one from the other side. Has a kinetochore microtubule from both sides of the cell. All right. And this, at this point in the game, is a very pretty part in a microscope. You can actually see this in a microscope. What you would see, and I'll try to draw a microscope drawing for you underneath here. So what you would see is this like really, really dark line here, because you can't see the DNA or the chromosome structures, but you would see like this, you know, fuzzy line in the middle. That's because all of these chromosomes, all 46 of them, are just like all kind of tucked into the middle here, and they're all lined up along their centromere. So the other thing you would see here are these like lines going in and out of it. You can't really see the centromere structures too well because it's a microscope, but you would see these lines going through it like that. That structure right there, once all of these guys are connected and everything's lined up in the middle, this whole thing right here from end to end, that's called the mitotic spindle. It's all these strings, if you will, creating this spindle-like structure. And these strings are the microtubules connecting to the kinetochores along the sister chromatids. So at this point in the game, the mitotic spindle, this lovely structure of microtubules reaching out to the chromosomes, that's what we call the mitotic spindle from end to end. The mitotic spindle is formed. This is the only time in mitosis that you're going to see the mitotic spindle. From here on out, in prophase and prometaphase, the spindle was starting to grow. At metaphase, it's complete, and pretty soon we're going to start going back in. So this is the only time you're going to see a beautiful mitotic spindle. All right, so that's metaphase. Okay, that might be one of my favorite phases in mitosis. It's all my favorite, I can't lie. It's all good. So big D, after metaphase is a phase called anaphase. So you can kind of picture what's starting to happen to the sweet cell here that we're drawing out, is that now that we've aligned our chromosomes in the middle, this is going to be big D, we got anaphase. So now that we've lined up our chromosomes in the middle, now we get to split them. So during anaphase, this is when it actually happens. All right, so I'm going to draw our centromeres here and our centromeres here. I'm not going to draw the chromosomes yet because I got to show you what it looks like. So if I'm a chromosome, I'm sister chromatids, I'm connected by my centromere right there, and I got my kinetochore microtubules attached to my wrist region right here. What's going to happen as these kinetochore microtubules, what we used to think as scientists in the field, that these centromeres used to reel in the microtubules. What we've since, and actually as they reel them in, they would pull the chromosomes to the opposite poles of the cell. What we now know is that there's a specialized protein in the kinetochore region that's actually chomping back the microtubule and so instead of being reeled in, they're being like, they're pulling themselves in, which is really interesting to think. So as this group of proteins, which we're not going to get into that detail, I just thought it was kind of cool, that these proteins that are pulling in and chomping away at the microtubules, they're pulling back the chromosomes as well. And so the chromosomes take this distinct shape. They're in this X pattern right now, and they're very much tied into the middle here by these kinetochore microtubules. As they're getting yanked to opposite poles of the cell, they go from this X to like this shape, like this. It's as if someone were to grab you by, your belt and like pull you, you're gonna go like this. That's exactly what's happening with the chromosomes. So the chromosomes that used to have this X shape now get split and they go into this kind of shape. So I will do my best. Oops, It's a little bit too, not as condensed as I want. So, it kind of, my students say they look like little hearts, and they're kind of right. Oop, I didn't figure that one out. Let's do that. All right, so now these kinetochore microtubules had it here, and now they're pulling them apart, and they're moving in that direction. And we also have these non-kinetochore microtubules, again, always working. So let's write this out. So in anaphase, here's what's going on. This is the shortest phase in mitosis, actually. So we just have the longest phase in metaphase, where it took forever to get everybody lined up. Now that they're lined up, you could basically say, okay, you go in that room, you go in that room. That's what's starting to happen. So during anaphase, the sister chromatids separate and migrate to opposite poles. So we had our sister chromatids, and now they're beginning to separate. So sister chromatids separate and migrate to opposite poles. One's going to this pole, one's going to that pole. All right. So there's these beautiful little motor proteins that are kind of breaking away the tubulin protein and the microtubules to just pull them back in. It's pretty kind of cool. During this whole entire time, as these guys are coming in, the non-kinetochore microtubules are actually getting longer and longer and longer and longer. So it's kind of like this back and forth. Like as this chromosome is coming in, these non-kinetochore ones are going out to lengthen out the cell even more. So non-kinetochore microtubules lengthen more. If I can spell it right. To stretch the cell. All right. You can kind of imagine where this is heading next. So now that this set of chromosomes are migrating over here, this set of chromosomes are migrating over here, if I were just to cut this cell in half, I would have two cells with identical DNA. That's the thought behind it, and that's exactly what's going to start happening. So that leads us into our next phase, which is called telophase. So in telophase, T-E-L-E, phase. In telophase, here's what's going on. I'm sorry, T-E-L-E, T-E-L-O. I'm saying it, writing it the way I say it. Telophase is, so now the cell is getting stretched out, and this is when you start to see the beginning of the cell division. And so what's happening is this one cell actually starts to become two cells, and it just looks like it's just starting to like pinch in the middle here. And at this point in the game, we have our centrosomes that were on opposite poles for a reason. They're going to be the sides of the two new cells, and at this time in the game, the microtubules are starting to shrink back in, things are happening, they're starting to help create what this whole area right here, this part right here is called a cleavage furrow. So a cleavage furrow is happening here, and it's, imagine if you're looking on the outside of the cell, what you would see is this cell that's separating and it's starting to curl in on itself, which makes perfect sense. If I'm going to be a big cell, I'm going to have to pinch myself in half to create two new cells. That's exactly what's going on here. So inside these two new almost developed cells, we need to rebuild the nuclear envelope. So the nuclear envelope is rebuilding. And remember, we don't have sister chromatids anymore. The sister chromatids got separated and moved to opposite poles. So now I have my two chromosomes here. They're allowed to float around here. but they're going to start being encased in the nuclear envelope that's forming right there. So for telophase, here's what you got to know, is that, remember, everything is in the feminine form when we talk about cell division. We had our parent cell, and now we're creating 2 new daughter cells. At this point in the game in telophase, 2 new daughter nuclei are beginning to form. So 2 new daughter nuclei, which is plural for nucleus, 2 new daughter nuclei are beginning to form. Nuclear envelope. I'm not spelling today. Envelope. Rebuild. And that last part is the cleavage furrow begins to form. And at this point, now that we have created 2 new nuclei, technically mitosis is complete. At the end of telophase, technically mitosis is complete. But we still haven't made 2 new cells, and that's where cytokinesis is going to come in. So the cleavage furrow forms. And number 4, mitosis is complete. So if you remember the definition for mitosis, it's division of the nucleus. We've divided the nucleus. We've split it up in 2. We had 2 chromosomes made of sister chromatids, and now in each new nuclei, we have 2 chromosomes made of 1 chromatid. Cool. So the count of the chromosomes in each nucleus hasn't changed. It went from 2 to 2, but it went from sister chromatids to single chromatids. The chromosome count has not changed. So we're still diploid in this cell. So lastly, let's do cytokinesis, and I'm going to keep that one on the board so we could talk about how, really quickly, cytokinesis is easy. So big letter F. Now we gotta split this guy. So letter F, cytokinesis. So in cytokinesis, this is when you actually get splitting of the cells. The cell gets made, nuclear envelope is there, you get your centrosomes, which are totally there. You get your chromosomes, they're doing their thing. Get your nuclear envelope, centrosomes. There you go. So now you have 2 identical new daughter cells. So cytokinesis, final step, that splits the cytoplasm to create 2 identical daughter cells. Nuclear envelope is completely rebuilt. And cells enter G1. They're now in interphase now. They're doing their job. And so that is, when we like to think of mitosis, that's when we usually like to think of the end of mitosis. Technically, it's fine if you lump these 2 together, telophase and cytokinesis. Totally fine. Everyone assumes that if you've entered telophase, you have to go to cytokinesis. It can't happen one way or the other. And so they're together. They're always together. So that, my loves, is cellular division. Let's recap really quickly. I'm just going to I'll keep this on the board really quick. So big G, we'll just do a summary of mitosis. So you have 1 parental diploid cell with sister chromatid chromosomes going to 2 identical diploid daughter cells with single chromatid chromosomes. Okay, that's mitosis in a nutshell. But all those phases are super important. You got to know about prophase, anaphase, metaphase, all that good stuff, prometaphase, all that. So I hope you enjoyed mitosis. And when you come back, we're going to start meiosis, and we'll talk a little bit more and get a little bit more intro before we get into that as well. All right, I'll see you all soon.